one of our best here, and still local with Novozymes. Come on up. Novozymes North America. And how long you been there? 5 years. 5 years? Okay, great. And she's a innovation manager, so she's got innovation to talk about. Thank you.
Thank you. I'm going to be talking about how to increase the value of fish co-products and waste. So continuing on the theme that we've started. More specifically, I'm going to be talking about how enzymes can help to increase the value here. And some examples are to make functional and flavorful protein extracts, to have better oil separation and higher quality oil, and have cleaner bones so that you can get a higher price in the supplement markets. I have an example process here, and this is just an example. So there are a lot of different companies doing it different ways. I'll go into a little more later about how you can change the process to get a tailor-made product. But this is just to start out of one way that you could do it. And so you would grind the co-products with water, and then add your enzyme, incubate at a specific temperature for however long you need to have the reaction that you want, and then have an inactivation step where you're heating up the product so that you are denaturing the enzyme. You then would go through a sieve to remove the bones. And this is a step you can stop here. So if you're going into something like the pet food market where you just want a liquid protein fat source that can be incorporated into the kibble, you can sell that at this point. Or you can take further steps where you're actually separating out the protein extract, the oil, and the sediment, and then selling those into different markets. And so you end up, after this process, with an oil layer, your functional protein extract, and then your sediment, which your sediment is just the insoluble protein, what you weren't able to get in solution. And this a lot of times still goes to the pet food market, whereas you've created more value in your oil and functional protein streams. And Novozymes is not the only one that's looking into this. There are quite a few lab groups at different universities that are looking into how to increase the value of the co-products and waste. One of the studies is shown here where they were looking at mackerel and how to make a high protein yield from the co-products. And you can see that they're hydrolyzing at different time points. And so there are different enzyme concentrations and then different time points. But most of them, you have this really high rate of initial reaction, and you're getting a lot of protein into solution fast. So this is a point where you can run for an hour, get a lot of protein in your solution, have a good soluble protein extract, and then you're not going to continue to get much more out. If you want to go further into a specialty product, though, you keep hydrolyzing. So while you're not getting any more protein out of solution or into solution, you are continuing to make smaller and smaller proteins. And this is a different university study that showed a similar result where, again, an hour you've gotten pretty much everything into solution that you're going to get into solution. But if you want to have more free amino acids, smaller peptides that might have more functional properties or health benefits, you can keep hydrolyzing until you get to that point where you want to have the product stop. There's also an effect of the protein hydrolysis time on the oil yield. So not only do you want to look at the protein component, but you're also looking at whether you can increase your oil yield by continuing to hydrolyze the protein. So here you have the 1 hour time point where you again have that high initial reaction, but you're seeing still some more benefit if you go up for another hour or two. It's not as flat of a curve as you continue to hydrolyze as your soluble protein yield. There are some critical process parameters that no matter what you're making, you want to be aware of. And the number one process parameter you want to control is temperature. And the reason for this is that enzymes can very easily be denatured. It's not like where if you put a chemical into a process, you can raise the temperature up by accident and then bring it back down, and maybe you still have the reaction that you were looking for. If you do that with an enzyme, you bring it up to the temperature where it denatures and then bring it back, you're not going to get the activity back. So you want to make sure that you're controlling your temperature. Most of the enzymes that we sell into this market, the proteases, have a temperature of around 55 degrees Celsius that they're most active for a long period of time. And if you go above 60, you start to lose that activity. It will vary by the enzyme that you're looking at, though. And then pH, you want to monitor your starting pH, and then you can actually follow the reaction to a certain extent by measuring the pH, because it will drop as you start to hydrolyze the protein. And if you're looking to separate the protein from the oil, you want to make sure that you don't accidentally emulsify them. So you have to be careful with your mixing and pumping so that you can actually get a good separation in the end. When you purchase an enzyme, you'll usually get a temperature curve, and most of the time it's something like this, where you're looking at the activity at various temperatures. That's the easiest assay to do, is to look at activity at the different temperatures. What you may be more concerned about, though, is the stability of the enzyme at the different temperatures. This first assay here, this is 15 minutes at each of the temperatures, but most processes run a lot longer than 15 minutes. So if you look at this, you may say, I want to run at 70 degrees Celsius because that looks like it's the peak for enzyme activity. But after 15 minutes, you're going to have close to no activity. So you're then losing out on that extra incubation time that you have. You want to make sure you get a stability curve as well. So this one is showing stability, where you can see that even when you get up to 60, you're starting to lose activity after 30 minutes. So if you're running for an hour, what's a better temperature is more like 50 or 55 degrees Celsius. And we're lucky enough to have some very nice customers who have allowed us to show some of their data. And so here we have some information about what you're likely to get from combining 1,000 kilograms of fish co-products with 1,000 kilograms of water. And this is one of our customers' typical yields, so it is averages. And one of the main differences you'll notice is that you have to have the right fish to be able to get your omega-3s. So your oil content of something like salmon is going to be much higher than other fish. And then your protein content is going to be a little bit different too, depending on whether it's a fatty fish or a fish that just has more protein. Usually when you get the protein extract out, you get about 5% protein in solution. So a lot of people will concentrate this material to closer to 30% because that's what the market is looking for, is a more concentrated source of protein. And one use for this fish protein extract is actually in the same facility that if you want to make it and add it to your fillets. So here you're directly impacting the water holding ability of your fillets. And this is an example showing an untreated cod fillet and then one injected with a fish protein extract. And you can see that after injection, you have a higher weight and you still hold on to some more water with that protein. So this is not pure protein you're injecting. That's why the weight drop looks maybe a little more than you would think, going from 114 to 107 after freezing. This is a 30% protein solution. But you're keeping that protein in the fillet and it's holding on to water and creating a more savory flavor. If you keep hydrolyzing beyond that 1 hour time point and you're making all those amino acids and peptides, you're actually adding a marinade-like product to your fillet here. And the consumer, when they thaw it at home, it looks like a bigger piece of fish. It has a higher weight, even though some of it is protein that's been added back. When you use enzymes in the process to make fish oil, you're also getting a higher quality oil. So a lot of times you'll have the typical fish oil process and you'll start to get a lot of free fatty acids. And if you quicken that process by using the enzymes, you're able to eliminate the free fatty acid generation and get a higher quality product out. You can sometimes get a higher total omega-3 content as well. And you have a nicer color. So I wasn't sure if it was all that important, but our customer shared with us that they really liked the color of the product. It was this reddish orange that you see up at the top, whereas with their traditional process, they were seeing just a really dark product that was not as appealing. We have several different products that we sell into these applications at Novozymes. We have alkylase, which is a very broad protease, and the broad specificity means that it just recognizes a lot of different cleavage sites. You'll get very small peptides if you continue to hydrolyze over time with this product. And I'm showing the optimum pH and temperature conditions here. So the range for those temperature conditions relates back to those graphs I was showing where 55 is where you would want to be for stability, but the true optimum temperature for activity is 70 degrees Celsius. So if you wanted a very quick process and just hit it at 70 and hydrolyze, you can go there. Or if you want to have more of a reaction and you're going to incubate for longer, you want to stay on that lower end of the range. And then we also have Nutrace and Protomix, which will give different cleavage patterns. So you'll get a slightly different flavor to your final product. They won't hydrolyze as extensively as the alkylase, but you'll liberate different amino acids and peptides. And then we also have an exopeptidase flavorzyme, which, as the name denotes, it helps to improve the flavor. You're cleaving off some of the hydrophobic amino acids that end up in the terminal end of the peptide and can sometimes cause bitterness. And then you're also generating more free amino acids and dipeptides that give you those savory flavors. And I would like to take any questions that you might have now.
That last cookie's weighing on me.
When we do research with some of these enzymes, sometimes we're asked, what are the actual enzymes as opposed to some trade names? Do you release that information, or is that considered proprietary?
We can give general information. I mean, we can tell you the organism, the fermentation organism, the host organism,
No, I mean, what do you actually know? Are most of these a mixture of enzymes, and do you actually know what the normal enzyme classification would be for those enzymes?
We can give you the EC number for them. Is that what you're looking for?
Yeah.
Yes.
Okay.
So that's information we share.
Good.
But these all label as, do they label?
It depends on.
Or you're talking about the finished product. The finished product obviously labels. Well, for example, in the fish where you're injecting the protein, how do you label that?
So you can say fish protein extract.
Fish protein extract is the. Have you had that approved somewhere? Or is that?
Well, we don't approve that. So
have
the
you
company
had it
that
approved
is
somewhere? Yeah. Do you know? Is
We-
it an
we
FDA
make-
or USDA?
we make a recommendation, and it's up to the company that is actually
Okay.
making the final product
All right,
to label it how they want.
Okay. Good.
But we do have customers saying fish protein extract, and the enzyme is not listed. It's a processing aid.
Okay. Okay. So it's always as a process aid.
Yes, and something like dietary supplements, it's not a processing aid. They actually want the enzyme on the label. But in this case, it falls
And on
under
the
the
label,
definition
it
of
just
processing aid.
labels enzyme?
You can choose to put the class of enzyme or just enzyme.
Okay, good. Anyone here?
That raises a question why it's called fish protein extract, because you're not actually extracting native proteins. It seems like it would be called fish protein hydrolysate, since you're hydrolyzing it. Or
Well,
is
that
this
would
just
be up.
semantics?
That would be up to the company that is making it. We can.
Yeah.
Well, yes, the company that's making it to understand the law and choose how they want to label it. I just know one of our customers is doing fish protein extract. I'm sure hydrolysate is another term that you could use.
Okay. Well, thanks, Katie. Thanks very much.